Hello, my name is Molly, and this is my presentation on our biosensor lab. So brief explanation of how this works. The two plasmids we're working with are the P dusk, which reacts by turning the reddish pink in dark environments, and then the P dawn, which also turns red and pink in light environments. The RFP, or red fluorescent protein cells, both react differently to the light. And then these systems are activated by blue light, also white light, and can change according to what light intensity and temperature you expose the bacteria to. So my hypothesis and process, if I expose my plates to the same temperature but to the opposing light intensities, then the RFP will not be as strong, but it will still be present. There will still be growth there. So essentially I wanted to see what would happen if I exposed the RFP to the opposing light intensities. So for example, what would happen if I inoculated the P dawn plasmid in darkness and the P dusk plasmid in light. So to start, I set up the 4 plates in the same room temperature, and I used one plate of the P dusk as my control unit, and inoculated it in the appropriate brightness. And then the other plate of P dusk was placed on a windowsill in the light, and that was my experimental group. And then I did the same with the procedures for the P dawn, but with respective light differences. So I had a total of 4 plates, 2 being the control plates, and then 2 being the experimental group. So pictures of the process. On the left here, I have this plate right here is actually a P dawn plate. I mislabeled it, but it is P dawn in the darkness, and then this plate right here is the P dusk in the darkness. Again, this one I just mislabeled. But then on the picture to the right, we have the P dawn in the light on the windowsill, and then the P dusk on the windowsill in the light. So results, the plate placed in the dark with P dawn grew and had the smallest amount of pink light, which was a little interesting. And then the plate placed in the sun with P dusk had little to no growth at all. And then the other plates placed in the correct light intensities grew fairly normal and had a small bit of pink tint to them. So here's the pictures. We have P dawn in the light, which as you can see has some weird growth patterns in it, but I know the pictures aren't really great. It is a little bit of pink here though, and that's the P dawn in the light. And then the P dusk in the light had no growth, hardly at all any growth. And then P dusk in the dark has a fair amount of even growth, and as you can see, I don't know if you can tell, but it does have a little bit of a pink tint to it. Again, camera quality is not super great here. But then here's the P dawn in the dark, which I was a little surprised that it had this much growth in it. Not a lot of the pink color, but I don't know, it's tiny tint right here. So in conclusion, my hypothesis was not entirely correct. There was, to my surprise, a bit amount of growth, and like I said, the tiniest hint of a faint pink color on the plate of the P dawn located in the dark. However, the plate of the P dusk placed in the light had nearly no growth at all. So if I were to do this experiment different again, I would definitely give myself more time to be a little bit more creative in experimenting with the light intensities. I could also add a lot more variables such as temperature. I could possibly see if humidity would affect anything, or I even had a thought if I possibly froze the plates and kind of, I don't know, preserved the plasmids somehow in the freezer, but then took them out and see if they grew again. I don't know if that makes much sense, but anyway, there are a lot of different ways to experiment, and like I said, if I had given myself more time and if I had thought about it a little bit more, I definitely would have tried some of these variables. I also would have made better taken better care to make sure that my plates were stayed clean, I guess, made sure that I didn't expose them to bacteria outside of what I wanted to inoculate. I guess that's just something I need to get better at. But anyway, I thank you for watching, and this was a pretty fun little lad to do.